I just realised I was recording things wrong. I have a pausing option on here, and I paused it, and I forgot to press play again. So, I just had to stop the video and see where I was. Anyway, rough endoplasmic reticulum. So the ribosomes are attached to flattened cisternae, and they transport proteins. Smooth endoplasmic reticulum. No ribosomes and tubular cisternae, and it synthesizes and transports lipids. So, and then you have ribosomes. They can be found in animals and plants, and they are attached to the rough endoplasmic reticulum or free in the cytoplasm. Ribosomes are smaller in prokaryotic than eukaryotic cells. In prokaryotic cells, they are 70S. S is just a type of unit of size, you don't need to know it. Whereas those in the cytoplasm of eukaryotic cells are 80S, where they occur singly or attached to membranes on the rough endoplasmic reticulum. Ribosomes have one large and one small subunit. They are assembled in the nucleolus from ribosomal RNA, so rRNA, and protein. They are important in protein synthesis, as they are the site of translation, where messenger RNA is used to assemble the polypeptide chain. Ribosomes are much smaller than the nucleus and mitochondria. They often appear as black dots in the electromicrographs, and in diagrams are shown as this. So you have one large subunit which contains 2 tRNA, which is transport RNA. Sorry, not transport, transfer RNA. So large subunit contains 2 tRNA attachment sites, and small subunit is mRNA attachment site, so that's messenger RNA. Now the Golgi body, or Golgi apparatus, or Golgi complex, in animals and plants. So the structure of the Golgi body resembles that of the structure of the endoplasmic reticulum, but it's more compact and is not interconnected. Vesicles containing polypeptides pinch off from the rough endoplasmic reticulum and fuse with the stack of membranes which constitute the Golgi body. Proteins are modified and packaged in the Golgi body. At the other end of the Golgi body, vesicles containing the modified proteins are pinched off. These may carry proteins elsewhere in the cell or move to and fuse with the cell membrane, secreting the modified protein by exocytosis. So, the functions of the Golgi body include producing secretory enzymes packaged into secretory vesicles, producing carbs for the formation of plant cell walls, producing glycoprotein, transporting and storing lipids, and forming lysosomes containing digestive enzymes. So you have the cisternae here, the incoming transport vesicle, the lumen, secretory vesicle, and newly forming vesicles. Lysosomes are in animals and plants. They're small temporary vacuoles surrounded by a single membrane, formed by being pinched off of the Golgi body. They contain and isolate potentially harmful digestive enzymes from the remainder of the cell. They release these enzymes when the cell needs to recycle worn-out organelles. The enzyme in lysosomes can also digest material that has been taken into the cell. For example, lysosomes fuse with the vesicle made when a white blood cell engulfs bacteria by phagocytosis, and their enzymes digest the bacteria. Some cells are programmed to die in a process called apoptosis, such as in the spaces between fingers and toes in the embryo. Lysosomes and their enzymes have an essential role in this. Centrioles are only in animals. They occur in animal cells, and most protoctistans. Most protoctistans, but not in the cells of higher plants. They are located just outside the nucleus. Centrioles are 2 rings of microtubules, making hollow cylinders positioned at right angles to one another. Together they are sometimes called centrosome. During cell division, centrioles organize the microtubules that make the spindle fibers. So vacuoles are in animals and plants, but they're different depending on what you're in. So most plant cells have a large permanent vacuole, which consists of a fluid-filled sac bound to a single membrane, the tonoplast. Vacuoles contain cell sap, a solution which stores chemicals such as glucose, amino acids, and minerals, and may store vitamins and pigments such as in oranges. Vacuoles have a major role in supporting soft plant tissues. Animal cells also contain vacuoles, but unlike plant cells, they're small, temporary vesicles and may occur in large numbers. Plants have small temporary vesicles too, but they also have large and permanent ones, but animals don't have those. Cell wall is only in plants. The cell wall of a plant consists of mostly cellulose. Cellulose molecules are held together in microfibrils, which are aggregated into fibers embedded in a polysaccharide matrix called celled pectin. The cell wall has the following functions. So transport. The gaps between the cellulose fibers make the cell wall fully permeable to water and dissolved molecules and ions. This space outside the cells through which solution moves is called the apoplast. The apoplast pathway is the main way that water crosses the plant root. Mechanical strength. The structure of the cellulose microfibrils and their laminated arrangement make the cell wall very strong. When the vacuole is full of solution, the cell contents push against the cell wall, which resists expansion, and the cell becomes turgid, supporting the plant cells. There's communication between cells. Cell walls have pores called pits, through which strands of cytoplasm called plasmodesmata pass. The plasmodesmata occurs where there is no cellulose thickening between two cells. The strand of cytoplasm runs from one cell to the next. This network of cytoplasm in the connected cells is called the symplast. The symplast pathway is important in water transport through a plant. So you have the cytoplasm, which is here, the endoplasmic reticulum, the cell wall, and the cell membrane. So the differences between plant and animal cells. Animal cells, no cell wall, no chloroplast, no plasmodesmata. Vacuoles are present but small and temporary throughout the scattered throughout the cell. They have centrioles and the energy stored as glycogen. Plant cells have cell walls, chloroplasts, plasmodesmata. The vacuoles are present and they are large, permanent, and central, filled with cell sap. No centrioles, but their energy store is starch. So organelles are interrelated. Organelles have been described here separately, but their functions within the cell are often related. So the nucleus contains chromosomes in which DNA encodes proteins. I have lost the lid for my highlighter. Nucleopores in the nuclear envelope allow messenger RNA molecules transcribed off of the DNA to leave the nucleus and attach to ribosomes in the cytoplasm or on the rough endoplasmic reticulum. I found my highlighter lid. Ribosomes contain ribosomal RNA transcribed from DNA located at the nucleolus. Protein synthesis occurs at on the ribosomes, producing proteins in their primary structure. Polypeptides are made on the ribosomes and are moved through the rough endoplasmic reticulum and packaged into vesicles. The vesicles bud off the rough endoplasmic reticulum and carry the polypeptides to the Golgi body, where they are chemically modified and folded. The Golgi body produces vesicles containing newly synthesized proteins. These may be lysosomes, containing digestive enzymes used within the cell. They may be secretory vesicles, which carry the proteins to the cell membrane for exocytosis. Phospholipids and triglycerides move through the smooth endoplasmic reticulum to various destinations in the cell. The cells of prokaryotes may resemble the first living cells. The oldest fossil prokaryotes are from rocks formed 3.5 billion years ago, so they must have evolved before then, within the first billion years of Earth's history. The cells of eukaryotes probably evolved from prokaryote cells, and the oldest fossilized examples are from rocks about 1.5 billion years old. Fungi, Proctoctista, plants and animals all have eukaryotic cells. Viruses are not made of cells and are not classified as living organisms. They seem to exist, the interface between living and non-living systems. So prokaryotic cells. An example of a prokaryotic cell is bacterium. The major distinguishing feature of prokaryotic cells is that they have no nucleus, or internal membranes. So, unlike eukaryotic cells, they have no membrane-bound organisms. In some prokaryotes, infolding of the cell membrane in a mesosome or photosynthetic lamellae increases the membrane surface area. They are unicellular and rarely form multicellular structures. The table summarizes their appearance. So, the diagram here: 70S ribosomes, mesosome, which is the site of aerobic respiration, DNA, which is free in cytoplasm, cell membrane, cytoplasm, cell wall, slime coat in some bacteria, plasmid, circular loop of the DNA, and the flagellum, which is... So this here is the flagellum. So all prokaryotes have DNA molecules loose in the cytoplasm. Peptid glycan, cell wall, 70S ribosome, cytoplasm, cell membrane, mesosome. Some prokaryotes have slime coats, flagella, photosynthetic lamellae holding photosynthesis pigments, mesosome, which is the site of aerobic respiration. It's also in all prokaryotes, and plasmids. So there's comparisons of eukaryotic and prokaryotic cells. So prokaryotic cells are small, so 1 to 10 micrometers. Eukaryotic cells are larger, so 10 to 100 micrometers. I've done things wrong. So in this table, that should say organelles. Prokaryotes have none. Eukaryotes have membrane-bound ones. DNA in prokaryotes is free in the cytoplasm. Eukaryotes is combined with proteins in chromosomes. Nuclear envelope. The prokaryotes have none. Eukaryotes have a double membrane. Plasmids may be present in prokaryotes, but they're absent in eukaryotes. Cell walls. So this one's made out. Prokaryotic cell walls are made out of peptidoglycan. And eukaryotes' cell wall is made out of cellulose in plants and chitin in fungi. Chloroplasts in prokaryotes there are none, but they may use photosynthetic lamellae for photosynthesis. And in plants, in eukaryotes, in plants, and some Protoctista, they have chloroplasts. Mitochondria, none in prokaryotes, but they may use mesosome for aerobic respiration. There are mitochondria in eukaryotes. There's mesosome present in prokaryotes and none in eukaryotes. And ribosomes in prokaryotes are 70S and free in cytoplasm. In eukaryotes, it is 80S and free in the cytoplasm and attached to rough endoplasmic reticulum. Viruses are small, so they can't be seen in a light microscope. They pass through filters that can trap bacteria, and although experiments in the late 19th century suggested their existence, they could not be seen until the electron microscope was invented. They are not made of cells, so they're described as acellular. There's no organelles, no chromosomes, and no cytoplasm. Outside a living cell, a virus exists as an inert virion. However, when they invade the cell, they are able to take over the cell's metabolism and multiply inside the host cell. Each virus is made up of a core of nucleic acid, either DNA or RNA, surrounded by a protein coat, the capsid. In some viruses, a membrane derived from the host cell surrounds the capsid. So here's the genetic material. This is the protein coat, and these are little spikes. Cells of all groups of organisms can be infected with viruses. The viruses that attack bacteria are called bacteriophages. A well-known bacteriophage is T2, which attacks the Escherichia coli, E. coli. Viruses can be crystallized, not a property associated with living organisms. The only characteristic of life that viruses show is their ability to reproduce, but they only do that when they invade a host cell, which contributes to the long debate as to whether they can be regarded as being alive. The new field of paleovirology examines the evolution of viruses. Paleovirology. They probably emerged several different times very early in the history of life. So here are examples of some diseases from viruses. So in humans you have flu, chickenpox, cold, HIV, mumps, rubella, Ebola. In plants you have tobacco mosaic virus, cauliflower mosaic virus. In birds you have avian flu. In other mammals there's swine flu, cowpox, and feline Cucamia virus. Levels of organization. So, differentiation and specialization. Single-celled organisms carry out all life functions within a single cell. Multicellular organisms have specialized cells forming tissues and organs, which have various structures and roles. Stem cells have the potential to become any cell type in the body. The development of a cell into a specific type is called differentiation. As they differentiate, cells become specialized in structure and in the chemical reactions they perform. Tissues. Cells near each other in the embryo often differentiate in the same way and group together as a tissue. Some cells in the adult remain unspecialized and are stem cells, with the potential to make any new cell type, like stem cells in bone marrow, can differentiate into all types of blood cell. Mammalian tissues. Mammals have several types of tissues, which include epithelial tissues, muscular, and connective. Epithelial tissue forms a continuous layer covering or lining the internal and external surfaces of the body, like your skin or in your gut. Epithelia have no blood vessels, but may have nerve endings. The cells sit on a basement membrane made of collagen and protein, and they vary in shape and complexity. They often have a protective or secretory function. The simplest form is cuboidal epithelium. There's a little diagram there, in which cells just have a cube shape. That's why it's called cuboidal. And the tissue is just one cell thick, so it's simple. It occurs in the proximal convoluted tubule of the kidney nephron and the ducts of salivary glands. Columnar epithelium has a long, elongated cells, so they're like columns. Those lining tubes that substances move through, such as the oviduct, which is in the fallopian tube, and the trachea, have cilia. So these are the cilium, and that would be a ciliated columnar epithelial cell. The nucleus, and this is the basement membrane. And then the squamous epithelium, which consists of flattened cells on a basement membrane. They form the walls of the alveoli and line the renal capsule of the nephron. So they're just a bit wiggly, aren't they? Yeah. Ignore these are silly notes, because it looks like Stollen. And that looks like the marzipan in Stollen. If you're familiar with it, it is quite nice. I like marzipan though, and sweetbreads. Muscle tissue comes in three main types with slightly different structures and functions. Skeletal muscle is attached to bones and generates locomotion in mammals. It has bands of long cells or fibers which give powerful contraction, but the muscle tires easily. You can choose whether or not to contract these muscles, so they are called voluntary muscles. Because you see stripes on them in the microscope, they are also called striped or striated muscle. So that would be these ones. You can see the stripes. Smooth muscle has individual spindle-shaped cells that can contract rhythmically, but they contract less powerfully than skeletal muscle. They occur in skin, in the walls of blood vessels, and in the digestive and respiratory tracts. You can't control these muscles, so they are called involuntary muscles. They do not have stripes, and so are called the unstriped or unstriated, and they don't tire, so that's what they look like. Cardiac muscle is only found in the heart. Its structure and properties are somewhat in between skeletal and smooth muscle. The cells do have stripes, as you can see. but lack the long fibers of skeletal muscle and smooth muscle. The cells have, yeah, I already said that. They contract rhythmically without any stimulation from nerves or hormones, although these can modify their contraction. Cardiac muscle does not tire. So, connective tissue connects, supports, or separates tissues and organs. It contains elastic and collagen fibers in extracellular fluid or matrix. Between the fibers are fat-storing cells, which are called adipocytes, and cells of the immune system. Blood, bone, and cartilage are also classified as connective tissue. Oh, I'm hitting the camera. Organs. An organ comprises several tissues working together, performing a specific function. In humans, for example, the eye contains nervous connective muscle and epithelial tissues and is the organ of sight. In plants, the leaf contains epidermal tissue, vascular tissue, and packing, or ground tissue, between the vascular bundles, which is specialized with photosynthesis. So, an organ system is a group of organs working together with a particular role. Drop my pen. Some examples of mammalian organ systems are shown in the table. So, digestive, stomach and ileum, excretory, kidney and bladder, skeletal, cranium, femur, circulatory, heart, aorta, reproductive, ovary, testes, respiratory, trachea, lungs, and nervous, brain, spinal cord. Organisms. All the systems of the body work together, making an organism, which is a discrete individual. And that is, as a matter of fact, the entire topic. I can go through some definitions if you want. So an organelle, a specialized structure with a specific function inside a cell. Prokaryote, a single-celled organism lacking membrane-bound organelles, such as a nucleus with its DNA free in the cytoplasm. A eukaryote is an organism that contains cells that have membrane-bound organelles, such as the nucleus, with DNA in chromosomes within the nucleus. A tissue is a group of cells with the same structure and function working together, and an organ is a group of tissues in a structural unit working together and performing a specific function. That is topic about cell structure and organization.